So I'm excited to talk today about the work we've been doing at LabGenius using avidity-driven selectivity mechanisms coupled with machine learning to overcome on-target but off-tumor toxicity events that we often see in T-cell engagers. So as many people in the room are probably familiar with, T-cell engagers are an extremely promising therapeutic space, and previous talks have gone at this as well. But many of these drugs are falling out due to on-target but off-tumor toxicity events. So we're seeing many drugs where they have strong and highly potent activity against the cancer cells, but against healthy cells that might express a relatively high level of the anti-TAA, you're also seeing relatively high levels of potency, and you have a very limited therapeutic window. There are some approaches to selectivity enhancement which are showing some promise and opening up a therapeutic window between these cancer and healthy cell populations. But this is still just that, only a window. What we're seeing with the platform that we've been building at LabGenius is an ability to get that complete level of potency against the cancer cells while having no detectable activity against the healthy cells. So we go from having a therapeutic window to having complete on-off killing selectivity with the platform that we've been building at LabGenius. So I'm going to start the talk by describing that platform in a bit more depth. And then I will go into case studies of the actual therapeutic spaces that we've applied these. So we call our platform the EVA platform. And LabGenius, we've been building this platform out of London. We have our facility where we've integrated our automation with the cloning, expression, purification, high throughput cell-based functional assays, and machine learning teams. Core to our approach is that our platform is really focused around this unique question of how do we optimize multi-specific molecules. This means we're thinking about the molecules themselves from a different way than I often see others in the broader machine learning antibody space thinking. We're looking at our multi-specifics as a combination of different building blocks. These building blocks obviously include the binders, but for these binders, we're not looking for sub-picomolar sorts of binders. We're looking for diverse panels of binders. So for both our TAA and our anti-CD3s, for example, we want a broad range of epitope coverage. So we want binders that hit the distal domain, hit the membrane proximal domain. We also want a broad range of affinities. We're usually looking for something like 3 log orders of affinity, from sort of sub-nanomolar to triple-digit nanomolar, sort of ideal for us. So the key here is not the most potent individual binders, but the broad and diverse range of those binders. Similarly, we're looking for broad panels of the other building blocks. So with linkers, we are looking at linkers of sort of short to long ranges and also varying rigidity, so more flexible and more rigid linkers. And finally, we add on a half-life extension domain to our molecules so that they're more therapeutically viable. We then assemble these molecules and evaluate them both computationally and experimentally for both function and developability. So in a typical problem space, we're going to be on the order of hundreds of thousands of theoretical designs of these multispecifics, usually T-cell engagers like I'll be showing today. And through the combination of the experimental systems and our computational systems, we're able to understand this fitness landscape for these vast number of designs. And ultimately, we end up with our optimized T-cell engager. And I want to dig a little bit into the specific format of these T-cell engagers. So just from eyeballing this, this is a clearly highly engineered format we're working with. We have our anti-TAAs indicated in red that are the building blocks that are binding to the target. These most often are VHHs, but they also could be scFvs or other sorts of binders. We have linkers connecting these different parts. And in green, we have the anti-CD3 that is attached to the FC domain in blue, usually working in high throughput mode with a single chain FC, but also not in all sorts of formats. There are some key benefits to this sort of approach we've laid out. The first is the selective tumor targeting. So by having multiple multivalent anti-TAAs, we're able to pursue an avidity-driven selectivity mechanism. With the way we've structured our molecules and the ways they can be arranged, this enables unique geometries. So we're showing here a C-terminal anti-CD3, but that can be moved around throughout the molecule. Because we have that monovalent anti-CD3, We reduce the risk of CRS compared to a format where you might have multiple copies of this. It's less likely that there's cross-linking of the T cells going on. The format itself and the building blocks are all amenable to subcutaneous dosing, which has obviously benefits when we actually look at the administration of these therapeutically. And finally, this FC domain gives us favorable properties from a half-life perspective, as well as manufacturability, because we can take advantage of standard sort of FC purification processes. And while I'll be focusing today on T-cell engagers, this platform we've been building is generally extensible to any multi-specific. We have active work internally around ADCs and are interested in opportunities to expand the platform to other sorts of multi-specifics, including TCR-based engagers like the previous speaker was talking about. A statement at the top here is obviously, it's going to be quite obvious to everyone in the room, that antibody discovery is challenging. We have a few specific reasons that we think of this as challenging. One is that we're trying to optimize on so many axes at once. So for an effective T-cell engager, you want something that's efficacious. So it's both potent, it's selective. You want something that's safe, so it has minimal tox and immunogenicity. It needs to be manufacturable, so you can express it at sufficient yields. It needs to be stable, so that it can actually be therapeutically administered to patients. The challenge is that these landscapes are often competing with each other. So the fitness landscape for efficacy might suggest molecules that ultimately are not thermostable. And we have to be optimizing over many different dimensions in terms of both the layout of these molecules as well as the readouts that we're looking at. And this is often hard for humans to understand and interpret. And that's where we start to leverage the machine learning due to the complexity of this task of simultaneously optimizing across so many different objectives. So the machine learning allows us to define really large really large theoretical design spaces indicated by the dark green box here. We have some throughput in the lab that allows us to subsample this space. And so we'll get sort of on the order of thousands of experimental readouts over this design space that's on the order of hundreds of thousands or millions. And so that's indicated by the middle plot here. But when we layer machine learning on top of that experimental data, now we're able to extract insights from neighboring designs in this space and understand more broadly this fitness landscape. So this very complex multidimensional fitness landscape of machine learning models over time, as we gather experimental data, are able to understand and learn from and propose molecules that are going to meet our ultimate criteria. So we're optimizing these models not only for the features of potency and selectivity, but also many features along the developability axes. To enable these models, we focused at LabGenius on building an iterative process that leverages active learning. So what I mean by that is we start off our programs with in silico designs. So we generate on the order of 760 to 2,300 designs in a particular cycle that we then, the dry lab team, which I lead, hands over to the wet lab scientists, where we do in-house cloning, expression, and purification of these molecules. We assay all of them to ensure that they're at sufficient purity. And then those antibodies get handed over to the assay teams, where you do high throughput functional cell-based assays, things like high throughput cytotoxicity assays, where we are working these up at scale and looking at them in multiple cell lines. The data from those assays then gets handed back over to the dry lab teams, where the models are updated, and then we leverage tools like multi-objective Bayesian optimization and batch active learning to propose that next round of designs that we then iterate on this process until we've achieved our TPP. One of the things that's often nice to highlight is that machine learning sort of the shiny jewel on the top of the stack, but this is really enabled by the ecosystem that we've been able to build at LabGenius. So while machine learning is what we'll often highlight the results from, this is enabled by the data generation processes that we have. So we focus on generating the most disease-relevant data that we can. For this question of selectivity, that means cell-based, high-throughput killing assays. We then have thought extensively about how we actually capture and store these data to ensure that all experimental data and metadata are stored in our systems that we're learning over time. The data are processed because fitting 2,300 molecules with dose response curves across 2 cell lines and 2 biological replicates of different PBMC donors in PRISM is not tenable. So we've automated the QC normalization curve fitting to enable this throughput. And then that allows us to do what I would consider to be the exciting parts of the active learning on top of this high quality data stack that we have built. So I'll now focus on some of the actual applications of the platform, which show the promise of this platform that we've been building. For our internal pipeline so far, we're focused on this challenge I've been talking about already of on-target off-tumor selectivity, on-target off-tumor toxicity that we're looking to solve through avidity-driven selectivity. So how exactly does avidity-driven selectivity work? Sorry. Okay, here. Sorry, the slides aren't working quite right for me. So the healthy cells express some level of this tumor-associated antigen, but a lower level than the tumor cell will. And so the receptor differences are highlighting that. Then we have the T-cell engagers whose formats I've already described, but we're focused here on the multivalency of that anti-TAA and the effects that that's going to end up having on the selective binding of the cells. So when we look at our healthy cell, this T-cell engager might have an interaction with the anti-TAs, but because this has been tuned to be slippery, it will fall off and not have a stable interaction. But in the case of the tumor cell highlighted here, the avidity mechanism will recruit engagement and lead to the ultimate recruitment of the T-cell and cytotoxic killing of that target cell. So we're looking for this avidity-driven mechanism to lead to that cytotoxicity event. When we look at the molecules we're actually designing, we have a lot of different axes that we're manipulating on. So in terms of the linkers, we're looking at different lengths of linkers and different rigidities. In terms of both the TAA and the CD3s, we're looking at molecules with different sorts of affinities. So as I mentioned before, sort of 3 log orders of affinities and different epitopes. And again, this is shown for the TAs here, but this is also true for the anti-CD3s. We're modifying the valency of the anti-TAA in these molecules and then the topology. So where exactly in that molecule does the anti-CD3 fit, for example? Our first case study was in the context of a HER2 CD3 T-cell engager. In this space, we had, so our first project, so we had a relatively small design space by our standards. We had 48 unique anti-HER2s we were working with that were mono to trivalent on any of the individual TCs. We only had a single anti-CD3 at the time, and we had 10 unique linkers of varying lengths that we were working with. This gave a total of about 44,000 designs when you take into account all the different arrangements of these various parts. And we assayed them at this point in our history on an activation assay. On the y-axis for these plots, you'll see that we have a T-cell activation score, and the x-axis, the selectivity score. In both cases, higher is better, so more towards the top right is an indicator of better performance. We have rinamotamab shown with the dashed lines as a benchmark. The plot on the right just looks at the top 25 molecules in any specific cycle relative to that benchmark on a compound metric that combines the potency and the selectivity. What we see when we look at our experimental data is in the first cycle, we have effectively a noisy distribution on selectivity and potency axes. But as the cycles progress, we see our molecules drifting more and more towards the top right. So over time, we're identifying more and more selective molecules. And to the point that by the 5th cycle, all of our molecules all of our top 25 molecules exceed that clinical benchmark in terms of performance. So we saw over time, the models were able to learn, propose more and more efficacious designs, and the cycle-based optimization was sort of showing its value in this context. I mentioned that the high throughput assay we had at the time was just an activation assay. We wanted to validate that there, in fact, was a killing selectivity profile that related to this activation profile we were observing. So we had rinamotamab as the benchmark molecule in the plot on the left in the blue. In both cases, the darker line is the cancer cell line equivalent, and the lighter line is the healthy cell line equivalent. You can see that with rinamotamab, there's some therapeutic window, but it's relatively small. And with our molecule, the top performer here, we had a level of potency that was equivalent to that benchmark against the cancer cell line, but no discernible killing of that healthy cell line molecule. And it wasn't the case where we just identified one of these molecules. We had a portfolio of many of these molecules that came out of our optimization process, where we had this complete on-off killing selectivity, where the healthy cell lines were never having levels of discernible killing. And the cancer cell lines had that complete potent and efficacious response. This was all really nice to see in HER2. It was also kind of an easy problem, if we're being honest, because there's roughly a tenfold difference in receptor density between HER2 and the cancer cells and the closest healthy cell line. So for our lead asset, we focused on a harder problem. It's a target we're not able to disclose just yet, but it's a TAA that has a threefold receptor density difference in a sort of typical cancer cell versus the highest healthy cell line that's known to express this target. For this problem space, we also increased the complexity of our designs. So we were looking at, in this case, 25 unique anti-TAAs. Now we had different epitopes that they were targeting because this is sort of a larger target. We were varying these from mono to trivalent again. We incorporate 3 different anti-CD3 flavors, including both VHHs and SCFVs. And we now had 20 different linkers because we had different rigidities in these linkers as well. So again, a design space of around 270,000 designs. And to cut to the end of this story first, when we look at the results, we see these same sorts of killing profiles. So in this case, there's not a relevant benchmark, but we do see on-off killing selectivity. The dark line, or the blue line here, is the high cell line, the cancer cell line equivalent, and the pink line is the healthy cell line equivalent. In this case, we're looking at about a 4-fold receptor density difference. It was one of the sort of higher tumor cell models we were looking at. But we then looked at an even smaller window for these same molecules of only a 2-fold receptor density difference. So the cancer cell line only has 2 times more receptors than the healthy cell line. And these properties are largely conserved. There's a slight decrease in the sort of Emax values on these curves, but overall we see activity against the cancer cells and no activity against the healthy cells, even in really small windows of for possible selectivity, which when we started this problem, we weren't sure if this would even be achievable with such a small delta there. So when we look at our overall portfolio, we're focused on developing these selectivity-enhanced multispecifics. I presented some of the data from our lead asset here. We have other TCs in the pipeline, as well as an ADC that we're currently working on lead optimization for. As I mentioned as well, we think this is amenable to lots of different formats, and we're interested if anyone would like to have a follow-up conversation around possible collaboration around any of these modalities. But that's all of the core content I have for today. Thank you all for your attention. I'd be happy to answer